Hello all, I am Dr. Varsha Shukla, Associate Professor in the Department of Microbiology of Ramnarayan Ruia Autonomous College. Today we are going to learn about a topic in microbiology titled An Introduction to Microbial Biofilms, where we will talk about the difference between planktonic and sessile cells. Then we will discuss about the significance of biofilms, advantages of biofilm formation, composition and structure of biofilms, stages of biofilm formation, multi-species biofilms and their role in nature and in infections. Then we will discuss about clinical biofilms, the different organisms that can cause biofilms in pathological situations, how these biofilms can be controlled or combated, and then lastly we will talk about some beneficial biofilms too. Although Anton von Leeuwenhoek had studied microorganisms or animalcules that he called them as from the tooth tartar, and therefore the organisms were surface bound and clustered together, for a long time microorganisms were studied only as planktonic cells. This was mainly because scientists like Robert Koch and Louis Pasteur and many other scientists later on studied microorganisms as planktonic cells in rich culture media. This led to scientists believing that microorganisms, the behavior of microorganisms in planktonic conditions is the same as the behavior in nature. However, later on, around 1970s, it became evident that microorganisms prefer to grow as surface bound, sessile, sedentary communities, lifestyles where microorganisms tend to cluster and behave as communities. These behavior of bacteria are much different than the behavior that they express as planktonic cells. With these studies, it became evident that biofilm is the normal mode of microorganisms growing in natural environments. In fact, planktonic mode is the mode where microorganisms grow only when the conditions are highly dilute natural environments.